All right, I also want to start by thanking the organizers for inviting me. This is quite a nice opportunity for me because this will be the premiere, the debut performance of a new molecule, basically, that we found in Vibrio cholera, a new communication molecule. And I'm really excited to share this data with you and hear what you guys think. So let me jump right in. And so you can see here on the screen while all these communication molecules are popping up, they are chemically actually very diverse, right? It's really hard to tell common structures. Some have long tails, some have different head groups. Even sometimes, as Karina already said, one molecule can change structure, can interconvert and make a new molecule. So from a chemical point of view, it's really hard to say what is a communication molecule and what is not. And so there are two molecules here which we know fairly well. This is AI2, which is used in Vibrio species, here with the boron. And this is CIA1. And these two molecules are used by our favorite organism, which is Vibrio cholera. And we know that CIA1 binds the CQSS receptor, while AI2 binds the LuxPQ receptor. And so what happens here, in the absence, at low cell density, if you want, in the absence of these two molecules, these two receptors, they function as kinases and phosphorylate these two proteins, first LuxU, which then transfers the phosphate to LuxO, and then importantly, LuxO functions as a transcriptional activator of 4 small regulatory RNAs, QR1 to 4, and these really perform the main functions of the chrome sensing circuit, first by repressing HepR, it's a major regulator of chrome sensing, and second by activating AFA, and that's basically the major regulator of single cell behavior, if you want. And so these two regulators really do a lot of things in the cell. It's a little counterintuitive in Vibrios because at low cell density through AFA you activate virulence and biofilm, while at high cell density HepR is actually repressor of both of these functions, right? Okay, so now let's go to high cell density. Now we have a lot of molecules here, and what happens, these two receptors, they change functions, they switch from kinases to phosphatases, and then what happens, QRR is no longer made, and then what will happen, HepR will be expressed, right? So this is what's happening. And so we were wondering, what are the global consequences of this, right? What is happening in the cell when it activates chrome sensing? And so we took a relatively simple RNA-seq approach where we have wild-type cells at low and high cell density, and we combined that with another little mutation that we like to use in the lab. It's called LuxOD47E, and what this does, it just constitutively makes the QRRs. So even at high cell density, where they're usually repressed, this mutant always produces the QRRs. So, and by combining all these mutations at, these mutants and doing RNA-seq of each of them at low and high cell density, we could come up with genes that are regulated just by growth, because nutrients change or whatever things happen during growth, or that are specifically regulated by chrome sensing through this pathway. And so, as I said, we did RNA-seq. We can do a lot of things. Expression profiling, I already indicated that. We can tell which genes are clearly chrome sensing dependent. We use the special technique in which we are able to identify the transcriptional start sites of all mRNAs in the cell and also of regulatory RNAs. So we can do an annotations of this transcriptional start site, and this allowed us to come up with something about 7,000 transcriptional start sites in Vibrio cholera. This allows us also to look at the untranslated regions of mRNAs, because when we know the TSS, we can define the 5' UTR of a gene, and often these are the sequences where post-transcriptional regulation happens. This is just shown here. It's a diagram showing you the average length of a 5' UTR in Vibrio cholera. Now, most importantly, and that was also my background, my previous background was, you can also use this really efficiently to find new small RNA regulators that haven't been found previously in prokaryotic genomes. And so, of course, we did that, and we came up with a list of about 107 new regulatory RNAs in Vibrio cholerae. And one was particularly interesting to us. Why? Because it wasn't annotated before and it's not obvious to find. So you can see here, this is an open reading frame. It's called VQMA. And VQMA is annotated to start here. That's where translation starts, and it ends here. But if we look at our RNA-seq data here, you can see that there's transcription start site here, which makes sense. That would give you a full mRNA, right? But there's another one here. And that would be odd, right? It gives you half a gene, half an mRNA, no translation of start site. That really wouldn't work. So it turns out these are actually two genes. This is VQMR, that's the regulatory RNA that we found here, and this is VQMA. So in comparison to the original annotation, this gene is just shorter, and it uses an alternate, another start codon to actually make the protein. We can also quantify that on northern blots. When we probe for VQMR, you can see here it's nicely expressed. Expression is low at low cell density, and then it increases towards stationary phase. It's not changed in the LuxO-D47E, and it's somewhat HFQ dependent, and I don't want to go into details here, but HFQ is an important chaperone for sRNA function. And so we wanted to go one step further, right? Now we have the regulatory RNA. We wanted to know what is driving the expression of that regulatory RNA, and our approach was as simple as possible. We have a transcription fusion to LuxZ in the genome of Vibrio cholerae. We perform TN5 mutagenesis. We screen for white colonies, and what we get is all hits in VQMA. So the transcription factor just downstream of VQMR drives the expression of this small RNA, right? It's a unit, they come together. And we can confirm that, of course, in wild-type cells, you have this typical expression pattern. You knock out VQMA, expression is completely gone, and we can compensate that on a plasmid. We can also show that in vitro by purifying VQMA, adding DNA from VQMR, and you get a nice shift. So it all works pretty well, pretty straightforward. I want to show you the small RNA that is involved here. So this is VQMR. VQMR has two domains that it uses for base pairing. One is called R1, and the other one is called R2. And we define targets of VQMR. I don't want to go into details because that might be a different story, but one target was particularly interesting to us, and that is called VPST. And VPST is important. You can see here in wild-type cells, this is the expression of VPST. We take out the small RNA, VPST goes up, and if we complement it, it goes down again. So VQMR is a repressor of VPST. And VPST is important in Vibrios because it controls biofilm formation. And so here's the network. You have chrome sensing coming in here from the top. Remember, this is the main regulator called HepR, and HepR represses VPST. And so, but when VPST is active, it actually is an important regulator because it activates these two operons here, and they make the glue for the biofilm. They make all the polysaccharides that hold the biofilm together. And so we tested the contribution of VQMR and VQMA to this scheme here, and you can see if we take out VQMR, you get about 2.5-fold increase in expression of VPST. You can take out HepR, and that gives you like about 5-fold higher levels. And if you combine those two, you get a really strong, right? So it's an additive effect. You have regulation at the transcription level and regulation at the post-transcriptional level. This is also then basically reflected by the activity of that promoter here, VPSL promoter. This is wild type. You take out VQMR, expression goes up. You take out VQMA, expression goes up, right? This is what you expect. We can also look at this under the microscope, look at biofilms, and you can see here, this is a strain we like to use. It's a HepR mutant, makes a lot of biofilm, and you know, like Dani said yesterday, we also have here cookies, but when we overexpress VQMR, we get to mayonnaise. So you can have both things. And when we add a point mutation, just one nucleotide that interrupts base pairing with VPST, we come back to cookies. So this works fairly well, and we know the small RNA works to repress biofilms in Vibrios. Now, this was all good so far. We wanted to get one step further and understand what controls VQMA, right? The transcription factor driving the small RNA. And that gave us a surprise, because I showed you before that expression of VQMR goes up towards stationary phase, right? OD.1, OD2 goes up. However, if you look at production of the transcription factor, it's constant, right? It's always there. And what that indicated to us is not the production of the transcription factor that changes, it's activity, right? And since we're working on chrome sensing, it made sense to check whether there's something in the supernatants that would activate VQMA. And so here's what we did. We have our control strain grown to low cell density, and then we just add cell-free soups of wild types, right? Same cell density, and expression of VQMR goes up. Now, of course, first thing to test, is it one of the two autoinducers that we know that play a role here, right? So we checked the activation in the LuxS strain, so they don't make AI2, but you can see it's still active, it's still activated, right? So it's not AI2. You can take out CIA1, it's not CIA1. However, and finally, if you take out both, right, it doesn't make a difference. So VQMA, our hypothesis is VQMA is activated by something extracellularly that is not CIA1 and that is not AI2. Okay, so now we had to go back to genetics again, and this time it became a little more complicated because we had to avoid cross-feeding on agar plates, right? If they sit really next to each other, we cannot really find the autoinducer because the neighbor would just secrete it. And so the way we did that, now we had a library, and we have the VQMR promoter fused to MK2, it's a red fluorescent protein, and we collected colonies, and then we asked a robot to sort them into 96 well plates for us, right? So it's all individual, so there's no cross-communication happening. And then we can screen these plates just on a multi-plate reader to see where we lose expression of this reporter. And that gave us just 2 hits, and these were in one gene that is called TDH, right? And TDH codes for threonine dehydrogenase. And here's the pathway. We heard about it yesterday already. So threonine is converted to aminocatibutyrate and then can eventually be converted to glycine or just spontaneously decarboxylate to aminoacetone. And so we test this. So we have a wild type here. This is the expression we get from our reporter. We take out TDH and we lose expression. We can take a wild type and introduce a TDH plasmid. Basically nothing happens, but if we take the TDH mutant and we complement it, we get back to normal levels. We also introduced mutations to the active site of TDH. So TDH is well studied. We know where the active site of that enzyme is, so we put 2 point mutations. And you can see here, this point mutation fails to rescue. There's a little bit of an interesting phenotype here. If you put this point mutation into wild-type cells, they actually also show reduced levels. And you have to know TDH functions as a multimer, and what we think this might be poisoned multimers, right? So there are multimers that are made of various components. And then we also wanted to make sure that this gene, the KBL gene, this one here, is not relevant for our purpose. So when we take the TDH mutant and we introduce the KBL plasmid, nothing really happens. Again, you see a slight reduction here in the KBL strain, and we think this might be because if KBL is overexpressed, it could basically drain aminocatibutyrate from here to glycine, which would then not be available for this part. Okay. Finally, make sure that KBL is not involved in the production of the molecule. So we have our TN5 mutant here, so we probe again on Northern blots for VQMR expression. TN5 mutant expression goes down for TN5 TDH. In the KBL TN5 mutant, you don't see an effect. So we think this part is irrelevant for the production of our molecule. Now, there are a couple of options now, right? TDH mutant, for example, could reduce the expression of VQMA, right? We had to check for this. So we did a simple experiment in which we just checked for VQMR levels and VQMA levels in wild type and TDH mutant, and you can see TDH shows the previously shown decrease in VQMR expression, but in VQMA expression doesn't change, right? So it's nothing about VQMA that is changing here, at least not production. Interestingly, we can take, show you before, we can take supernatants of wild-type cholera, and they activate the system. If we take supernatants from the TDH mutant, they fail to activate the system. And this is interesting. We can also do that with E. coli supernatants, right? So we can take E. coli, we get rid of all the cells, we add the supernatant, and we get activation of our system. And if we take a TDH mutant in E. coli, we lose that activation. So it's a conserved pathway that exists in enterobacteria that produces this molecule that we're looking for. Coming back to threonine, of course, there are 20 amino acids, and we checked for activation of our system in the presence of these amino acids, and you can see it really only works for threonine, right? That's the one thing that leads to induction. And basically coming from these experiments here, the question would be, maybe threonine is our autoinducer, right? That's possible. It could be just the amino acid. However, in this experiment, we can see where we take cells, we grow them to low cell density, and then we add either MOX, so that's just fresh media, or we add cell-free fluids from wild-type cells, or we just add a high amount of L-threonine, right? And you can see only the cell-free fluids actually lead to activation of VQMR and not threonine itself, right? And these are very short time points, right? Just 1 minute after adding the cell-free fluid, you get expression of the system. So we think it's highly dynamic. And so we were now basically left with the task to find that autoinducer. And we tried various strategies, and what finally worked is that we were lucky that our protein can be made in E. coli, because I showed you E. coli makes the molecule, right? So we overexpressed VQMA in E. coli to large amounts, so hundreds of liters of cells. We purified the protein and denaturated at a 50 degrees. So basically what happens, the protein captures the molecule, we heat it up, it folds open, and it releases the molecule. We purify that, and we send that for, we check that for activity. You can see here, this is, again, just an induction assay. This is our soups here, 25%. However, we need about 100 to 1,000-fold less of what is basically coming out of here. So we enrich for the signal, right? And so we did an important test here. So we took this and used that for NMR and mass spec experiments. And so this is our VQMA capture. And as a control, we used LuxR. It's another transcription factor in cholera that serves as our negative control. And we can see one nice peak here, right? So we went on and purified that peak. We can see, so now we know where we have to look, and we can see that peak is really abundant in wild-type cells, but it's missing in TDH cells. And based on that, we could actually identify the molecule. Now, basically, to prove the pudding, we had to take DPO, which decided to call it, is a dimethylpyrazine. And so we take DPO, we get it synthesized, and we add it to cells, right? And you can see here, this is wild-type cells, this is cell-free soups, and then when we add DPO and decreasing concentrations, we get a dose-dependent response. Again, making sure that DPO is really directly interacting with our transcription factor. We had the synthesized DPO in hand now, and we performed a similar experiment as before. We grow cells to low cell density. We either add threonine. You saw before, nothing happens in 16 minutes. It takes longer for this to happen. But if we add DPO, we get really rapid induction of the system, right? And similarly, if we add cell-free fluid, it looks very similar. So does it do anything on biofilm formation, right? This is where we're coming from. This is where we started. And the answer is yes. So this is a typical Vibrio colony, right? And it can form these wrinkles if you keep it on plates, right? So after a while, it forms these wrinkles. However, when we add DPO, this is clearly delayed. This takes much longer to develop these wrinkles. And we also developed another assay. This is based on some of the ideas that Melanie presented yesterday. This is a chitin bead. And Vibrio really likes to form biofilms on chitin. And so this is without treatment, and you can see it forms this nice clusters here on this bead. This is the TDA, this is the VQMR reporter. You see this is in a TDH mutant, so it's dark, and you can see this is basically the merged picture. However, if you add DPO, you find this is a chitin bead, but you really see that the cells are much more dispersed. They don't really adhere to the chitin bead anymore. So we think that DPO is another layer of repression of biofilm in Vibrio cholera. Now, is that important? Is that important to disease of Vibrio cholerae? And yesterday we already heard from Jean-Marc that there is an important molecule in the intestine of humans and mammals, and that is called mucin 2. And mucin 2 is interesting in terms of its threonine content, right? So this is mucin 2, and you find in the middle, so the central part really is really high in threonine repeats, right? So when you have wild-type cells and you add mucin, you really get a nice induction of VQMR expression. However, if you do that in the TDH mutant, nothing happens here, right? So Vibrio cholerae basically can, that's something I should say. Vibrio cholerae has the enzymes to digest mucin. It can get to it, right? It can use it as a carbon source. And in infection, it really likes to stick to this, right? And so when you add mucin to these cells, they start the system. And we were also encouraged by data from the lab of Jeffrey Gordon, and because he showed that when he looked at large data sets of cholera infections from Bangladesh, and he was looking at the microbiome, and he found during recovery from Vibrio cholera infection, he found that one species that is called Ruminococcus obeum became really abundant, right? So it's kind of a protective thing. They tried that. You can see this is their data. So if you add Ruminococcus to a Vibrio cholera infection, you really get a strong decrease of fecal content in Vibrio cholera here, right? If you take that black bar here. And so this motivated us to see whether species from the microbiota would actually participate in making that signal and making DPO. And in fact, we find if we co-cultivate here specifically Vibrio cholera with Ruminococcus obeum, we find strong induction of VQMR expression. However, this is not true for B seta or B fragilis, right? So what we think, there's a molecule that is made by the microbiota that is sensed by Vibrio cholera to repress biofilm during the infection process. And this is where I would like to end. I would like to thank the people involved. Most of this work was performed in the lab of Bonnie Basler. These are the chemists who really helped us with this project. The initial part was, the sequencing part was performed in collaboration with Cynthia Sharma at Würzburg, and this is my team in Munich, and I'm happy to take your questions. Thank you.